So after talking about what species are and applying definition, for example, the general lineage concept, to a few case studies like we did with the snapping shrimp, fruit flies on the hawthorn trees, etc. Let's move on to this topic of how rapidly speciation takes place. Indeed, Charles Darwin's seminal book, Origin of Species, his view was that speciation was a slow, gradual process, inevitably, that it would take a considerable amount of time for one species to truly diverge into two species. So now we're able to really see and address how rapidly speciation takes place. We have plenty of observational data, empirical evidence to answer this. So what we're looking at here is Coyne and Orr, who are two very prominent evolutionary biologists, rated reproductive isolation of 171 species pairs of fruit flies, Drosophila, with a score of 0 to 1. So if the two species that they paired up to see if they could mate were not able to have any offspring at all, they were 100% reproductively isolated, then they earned a score of 1. If they were able to freely breed without any impairment, no prezygotic barriers, no postzygotic barriers, nothing, then they get a score of 0, with scores all in between. So they also used a molecular clock approach to estimate how long ago the various species pairs diverged from one another, how long ago they shared a common ancestor. So in doing this, they're able to compare the score for reproductive isolation against the amount of time that we estimate based on DNA divergence or protein divergence, how long ago they shared an ancestor. And what they look at the figure here, genetic distance compared to reproductive isolation. So what they found is that it could take as long as several 100,000 years to become completely genetically isolated. So there was no specific amount of time that always popped up, that it takes X number of years for these fruit flies to become reproductively isolated to some degree. It's pretty variable, as you can see on our plot. And also, another interesting observation, and it makes sense, I think, if we think about it, talk about it a bit, prezygotic isolation evolved faster than postzygotic isolation. So arguably, postzygotic isolation probably wouldn't start evolving until a species pair began reproducing. There would be no selective pressure for, say, hybrid sterility or hybrid inviability until species began to attempt to reproduce with one another, or at least that kind of postzygotic isolation would be expected to accelerate once a species pair came together. But with prezygotic isolation, remember that there's temporal isolation, behavioral isolation, lots of different prezygotic things, and there's going to be selective pressures, arguably, for those kinds of things from the environment much earlier than postzygotic. So the upshot of this then is that it doesn't take a specific amount of time for speciation. Using a molecular clock approach with animals like this, with insects, we can get a gauge of how rapidly speciation takes place, and it's actually quite variable. So we just took a look at the rate of speciation using fruit fly example, comparing the amount of reproductive isolation to genetic distance by the molecular clock. And now we're taking a look at the rate or speed of speciation from another viewpoint, using especially plants with the phenomenon of polyploidy. And specifically here from our evolution textbook, allopolyploidy. You might recall, depending on what you heard, read, learned in your background, that there's also autopolyploidy. So here we're just looking at allopolyploidy. And allo means other or different. So like allopatric speciation, different fatherland. So here we have allopolyploid. And the allo here, the different, is referring to the fact that this occurs when two species hybridize. And they have different chromosome numbers that has evolved since their divergence, since their isolation, since they've been evolving independently. So, of course, they make their gametes, but they're still able to reproduce. Their gametes can still combine, fertilize, and we still have a viable offspring. So we get a hybridization event between species, and it would then result in most likely in a sterile hybrid, because the chromosomes that are being received from the two parents of the two species are not homologous chromosomes. They're different chromosomes. So essentially underlining here, they end up having at least some unpaired chromosomes. Now, if we look at the figure that's off to the right, just to kind of lead you through this up to this point, we've got a parent species with 3 pairs of chromosomes over here. We've got another parent species with 2 pairs of chromosomes over here. So when they go through the dance of meiosis, the organism, the species on the left, is going to do meiosis and make gametes with 3 chromosomes. And the other species, the other organisms, are making gametes via meiosis with 2 chromosomes. So when they mate and gametes are combined, we end up having this hybrid that is viable, but it is sterile, because if you look at the chromosomes in the cell of this hybrid, they are indeed unpaired. We've got 3 blue ones, single chromosomes, and the 2 red or pinkish ones from the other species. So, plants are exceptionally good at reproducing asexually. And that's how many of these hybrids are able then to persist and reproduce. They can reproduce asexually. And they can persist. They can grow. Now they're growing separately. They can't reproduce with the parents they came from because they have different chromosomes than the parents do. And they're essentially reproductively isolated immediately. But they can't reproduce sexually themselves because they have these unpaired chromosomes. Now, one thing that can happen, and there are various things that can happen, is that eventually one of these hybrid descendants can accidentally duplicate its entire genome. So as it's going through the dance of mitosis or even meiosis, the chromosomes don't separate properly and a genome gets duplicated. If that happens, then we're down at this situation near the bottom of the diagram where we now have an organism that has paired chromosomes. And now that that organism has paired chromosomes, it can now do meiosis and make gametes, and those gametes can combine and end up having a diploid, in this case on the very bottom figure, a diploid offspring that can now do meiosis. And now we've got a species that can sexually reproduce, has gained the advantages of sexual reproduction, and voila, we've got a new species. So this kind of speciation is virtually instantaneous because of this polyploidy event. Isolates the offspring reproductively from the parents. So we've got a fruit fly example where speciation takes on average a couple 100,000 years. And here we have a plant example where speciation is virtually instantaneous. The last example on the slide is with the genus Tragopogon. These plants that live in the Pacific Northwest, eastern Washington, Idaho, etc. There's native species there. Species were brought over from Europe in late 1800s, early 1900s. They began to hybridize. And with that hybridization, new polyploid species came about. So if you look at this diagram, I'm going to use my magic red pen here. We've got Tragopogon pratensis that has 2n equals 12. We've got Tragopogon dubius, 2n equals 12. And each of these donates a set of chromosomes. And now we eventually have this species 2n equals 24. So after the chromosomes duplicate. And then we have another example, Tragopogon dubius, which is in the middle, hybridizes with another species, Tragopogon porifolius, on the right. And we would first have hybrids that have unpaired chromosomes, and then eventually we might have this accidental duplication and Tragopogon mirus as 24 chromosomes. So yet another example, I mentioned this kind of thing earlier, where we have a theoretical possibility. This is what could happen with accidents in meiosis, etc., to produce, hybrid organisms that are reproductively isolated. This is a model for rapid speciation, and we have an empirical example. We have several empirical examples of how this has actually happened in nature. Under our watch, we have observed this over the past 100+ years. So we just covered the rate of speciation. Now we're moving on to another topic titled Uncovering Hidden Species. The bottom line here is, with the advent, invention, if you will, of molecular techniques, technology, sequencing DNA, and many decades ago being able to look at protein variation among populations and species, this led to the discovery that there's very often hidden species out there. Species that look the same morphologically. We have a difficult time distinguishing them from each other using typical anatomical characteristics or maybe even behavioral characteristics, but especially anatomical things that biologists often use to distinguish species. So let's go through this example and get to this concept. So this example, tons of examples out there in the literature of this kind of thing. Cute little butterflies shown down in the very bottom inset left corner of the slide. These butterflies called skippers, very diverse. We've got skippers in Arizona. They're pretty common. Astraptes fulgurator. So there's the species name. Very widely distributed species occurring from, if I remember correctly, Argentina, all the way up through Central America and into North America. So one single species, widely distributed, and the butterflies being morphologically, color-wise and other characteristics, not very different. But one thing that was known about these butterflies was the caterpillars in various localities, various geographic areas, feed on different plants. So that's a potential clue that there's something interesting going on there, at least ecologically, because of course plants commonly evolve chemical defenses against insects so they don't eat them. So if the caterpillars of these skippers are eating different kinds of plants, that suggests that there's probably some, considerable evolution that's happened to allow these caterpillars in different areas to be able to eat different plants that have different chemical defenses. So researchers did some genetic testing of these butterflies across their distribution from Argentina northward, and led to the discovery of at least 6 different species, distinguished from molecular genetic characteristics. So at least 6 different independently evolving lineages that we would say, aha, these qualify as species. So now we get to the term that we use for these kinds of species that are difficult or potentially impossible to distinguish like anatomically, but indeed they are genetically different and evolving independently. And we call these cryptic species. So recall that word cryptic essentially means camouflaged. So camouflaged species. We can't tell them apart visually. They look similar. There hasn't been arguably natural selection to favor them to evolve differences as adult butterflies, but natural selection has favored their larvae, their caterpillars, to be able to eat different foods. So biologists went on to then look at the species of wasps that parasitize the larvae of these caterpillars, because there's very often specific species of wasps that will parasitize specific species of, say, butterflies, lay their eggs on the caterpillars, and then the larvae of the wasps basically eat the caterpillars alive, and the larvae of the wasps then pupate and turn into wasps. So remarkably, they found that there were dozens of species of wasps that parasitized the larvae. And in fact, if I remember correctly, it was upwards of 300 and something odd species of wasps that they ended up discovering cryptically that were parasitizing these larvae. And maybe not all cryptic species of wasps, but the point is that discovering these cryptic species of butterflies led to the investigation of the parasites that live off of them, and that revealed that there are many, many more species of wasps as well that had been co-evolving with these butterflies. So cryptic species are observed in all kinds of lineages, from frogs to fish, birds, of course insects. You might also say plants that have potentially speciated by polyploidy could be cryptic. They're genetically isolated, reproductively isolated, but we maybe can't distinguish them. Cryptic species. So we've been looking at sexually reproducing species of eukaryotes, and we've talked a bit about how asexual species are also covered under species definition, like the general lineage concept or the phylogenetic species concept. So, but what about microbial, quote, species? What about prokaryotes, especially bacteria and archaea? How should we view species, or how do these definitions that we've talked about apply to microbes such as bacteria and archaea? Well, let's take ourselves back to the 18th century, where biologists were naming species of bacteria using Linnaeus's binomial system. And of course, they're only able to use observable traits that they could see through a microscope to distinguish bacteria, which is pretty limiting, as you know. So really, the biological species concept doesn't even apply to these kinds of creatures, as I've already mentioned at the intro to the slide, because they do not reproduce sexually. So instead, perhaps maybe a phylogenetic species concept is more appropriate for things like bacteria and archaea, since there's no sex with them. So what biologists did is use 16S ribosomal RNA as a way to distinguish different species. The reason being that 16S ribosomal RNA is found amongst all life forms, so it's very comparable across different lineages of prokaryotes and other things. And because traditionally named bacterial species tended to be about 3% different, and the members within the species 97% similar, biologists decided, hey, let's use that criterion of 97% similarity, and let's use it to try to identify different phylogenetic species of bacteria and archaea. Things that we can't tell apart, like anatomically, morphologically rather, but using this percentage of genetic similarity as a criterion, let's go out there and see potentially how many different phylogenetic species there are. So following that threshold, they looked at E. coli, and they found that old E. coli, different strains of E. coli, it's 3 different strains that are shown in the top left figure, had a certain number of genes that were shared amongst all 3 species. And the genes that are shared among all 3 of these species are called the core genome. The total number of genes that are found amongst all the species is called the pangenome. So they found, first of all, that there were many more, quote, species, phylogenetic species of E. coli that seemed to pop up because of this genetic criterion. Many, many more species that are sort of hidden out there. We could potentially think of them as cryptic species. So since then, biologists have been using this criterion to look at other strains of E. coli. In fact, they've looked at over 2,000 strains of E. coli, and they have found that out of those 2,000 strains of E. coli, there's just about 3,000 plus genes in the core genome that are shared amongst all of them. But there's something like 30,000 plus genes that are found in the pangenome. So what this is getting at is that there is a lot of diversity phylogenetically of E. coli out there that has been evolving independently to some degree. And so under this criterion, then using the phylogenetic species concept with this criterion of genetic similarity, there is a profound amount of undescribed, quote, species of E. coli. In fact, there are millions of them, of E. coli. If we extend this to microbes in general, then indeed on Earth we can expect that there are trillions of, quote, species of microbes. Mind-boggling. Yet, down on the bottom here, if we look at traditional species like Streptococcus pneumoniae, that lives in our airways, causes pneumonia, and a very closely related species, Streptococcus mitis, that lives in our mouths, so they are ecologically different, which is part of the big reason why they were identified as different species. They are 99% identical. So what does this mean? What does this imply? What that implies is that there are species of bacteria out there that are actually genetically very similar to each other, potentially only 1% difference, and yet ecologically they are living in different places. And so they are independent lineages. So the bottom line here is that using something like a biological or even phylogenetic species concept for microbes, for prokaryotes, maybe doesn't make a lot of sense. Maybe the biology of microbes is so fundamentally different from eukaryotes that we need a different idea. And you know that this has also helped to show us this concept of the core versus the pangenomes, that there are species of bacteria for which there are many, many different genetic strains, and fundamentally they share core genes amongst all of them, but collectively there's a huge number of genes that are found among all the strains, and that is the pangenome. So another thing to keep in mind when it comes to our friendly neighborhood microbes, bacteria, archaeans, is that horizontal gene transfer is rampant. It's all over the place. And so horizontal gene transfer makes the PSC, short for the phylogenetic species concept, non-applicable. Because remember, that species definition is saying that species are independently evolving lineages that are diagnosable. We can tell them apart. And lineages are vertical entities, reproducing over time and producing new generations. We refer to that as vertical evolution. So that phylogenetic species concept is focused on that vertical independent evolution. But with horizontal gene transfer, that really messes up that vertical evolution. It really kind of dirties and messes up any idea of a phylogenetic species concept. So, in fact, as is noted on here, other studies have showed that on average, 80% of genes in species studied of microbes experience horizontal gene transfer. And the figure on the left is getting to that, showing that. So instead of bacteria being neatly divided by species barriers, in fact, they are, sort of more ecologically divided than genetically divided. Clearly, there's genetics behind this, but maybe our focus should be more on the ecology of microbes to help us distinguish different species. So on the left, we see an evolutionary tree of 181 species of bacteria. And on the right diagram, horizontal gene transfer is shown by the colored branches, typo there. And so it's showing that, looking at these 181 traditional species of bacteria, horizontal gene transfer is rampant. So again, the overall point here is that a lineage-based concept like the phylogenetic species concept just might not apply to this kind of life on Earth because they have a different kind of biology than us eukaryotes. So maybe there's a different idea that we should have about what species are and how we identify them in this microbial world because of horizontal gene transfer. So having addressed this idea that the phylogenetic species concept, or maybe a general lineage species concept, it wasn't discussed like that in your textbook, really doesn't apply to microbes because of horizontal gene transfer. Maybe there's, as I mentioned on the last slide, a different species concept that we should have for microbes because of their different kind of biology. So Frederick Cohen in 2006, 15 years ago, argued that microbes instead undergo ecological speciation. And that's what our focus should be on, to be thinking about identifying and viewing species in the microbial world. So, for example, microbes living around hot springs in Yellowstone National Park form genetic and ecological clusters. And so, the bottom right is kind of showing this. So if we look at a microbial mat of prokaryotes, living on a surface, then we find ecologically that these microbes have divided themselves, not by choice, of course, but natural selection has carved them up like this to be living in different ecological niches. So vertically, we can have one species up here on the top in this mat, and notice how it's focused down on this layer. We're looking specifically on this layer and this whole vertical cross-section of a microbial mat with lots of different microbes living from bottom to top. And then below that layer, there's a different species that's adapted ecologically to a different niche in that mat. And so that's the argument here, is that lineages that are adapted to particular niches should be regarded as species. And natural selection is going to sort of prune species side branches as mutations originate, because they're not going to be well adapted to these niches. And so with that happening, any mutations that pop up randomly here and there in bacteria living in these ecological niches, overall, unless there's some kind of new mutation that really is beneficial, for the most part, those mutations are going to be weeded out. And what that'll do is maintain the strong genetic and ecological cohesiveness of bacteria in these niches. So that is the idea of using an ecological sort of species concept, ecological clusters, rather than a phylogenetic concept for microbes. So finally, what we're ending up here with, this is the last slide for the PowerPoint, chapter 13, is having discussed in this section that microbes are fundamentally different from, that is, prokaryotes from eukaryotes because of their mode of reproduction, general biology, that a lineage-based concept such as the phylogenetic one, that focuses on genetics, really might not be applicable because of horizontal gene transfer. So here we're taking a final look at a speciation model for prokaryotes, those kinds of microbes. I'm saying this because we might also generally say single-celled protists are microbes, but here in your textbook, microbes is being used to really talk about prokaryotes. So a speciation model for prokaryotes that takes into account horizontal gene transfer. So we're now combining together this ecological niche kind of species concept with the idea that horizontal gene transfer occurs. So let's really use this diagram, this figure that's here. So we have ecologically associated populations of microbes, and the arrows going up and down here are signifying that there's horizontal gene transfer going on among these populations of microbes. So genetically and ecologically, they're kind of cohesive. And so genes are being swapped and acquired through horizontal gene transfer from unrelated microbes at this point. And now, because of that, some of these lineages, some of these populations are becoming ecologically separated. And so we start to get here at this fork, at this divergence point, ecological separation. We now have microbes that have different sets of genes that are enabling them to live in different environments, and they're now ecologically separated, and they are evolving independently from each other because of their separate ecologies. And so down near the bottom of the slide, as groups diverge, okay, after we have this split and they're now evolving separately, natural selection is going to favor mutations that improve performance in each niche. Any mutations that happen that don't work favorably in these niches are going to be weeded out. And you'll get homologous recombination continuing in each group. So note that we still have these little arrows that are going back and forth in each group, so there's still horizontal gene transfer, but it becomes less and less common between groups. We don't see horizontal gene transfer going on between this pink lineage up here and this yellow one down here because they're occupying different niches. So they're just not encountering one another. And now we have different sorts of ecological species of microbes. So this is what we're ending with, is perhaps this is the best view of how in the prokaryotic world we should be identifying and thinking of species with what we've come to learn over the past few decades. And then we have a fundamentally different species concept, like the general lineage concept, that we are using in the eukaryotic world. So as you can see, wrapping up on this chapter, this whole species idea of what a species is is very colorful, has a colorful history, is not easy to pin down and put our fingers on, but by and large, biologists have moved into fairly comfortable positions across life as to what species are, and we have pretty good models of how species originate, whether allopatrically or sympatrically. We've come to learn that species are isolated from one another by a variety of mechanisms for sexually reproducing things, prezygotic, postzygotic mechanisms, and that species evolve at different rates, sometimes instantaneously, like with polyploidy, sometimes very gradually, just with traditional sorts of evolutionary processes going on. And so we have a very colorful chapter here about species and speciation as we continue with our evolution.